Bismillahirrahmanirrahim. So today we are going to discuss transcription in eukaryotes. Comparatively, in eukaryotic system, transcription is a very complex process. Just like the transcription in prokaryotes, the eukaryotic transcription mechanism is a very complex process, and many transcription factors are involved for the process of transcription to be carried out. So E stands for eukaryotic. Eukaryotic RNA polymerase has its own promoters and transcription factors. First step, which is going to be held for the transcription, which is the formation of different types of RNAs from DNA in eukaryotic system, is the chromatin remodeling. If you have a condensely packed chromatin structure, which is called as the heterochromatin, you have to convert in euchromatin first. This process of interconversion of heterochromatin is euchromatin, and euchromatin into heterochromatin is called as chromatin remodeling. This is done with the help of different types of enzymes which are known as HATs. HATs are histone acetyl transferases, which are these histone acetyl transferases basically add acetyl group in the heterochromatin structure, which is the condensely packed structure of the chromatin. What basically happens as histone acetyl transferases are the enzymes, so they must have proteins in them, and these positively charged lysine residues causes the binding with the DNA, which causes the decrease in histone and DNA interaction and converting the condensely packed structure of the chromatin into euchromatin, which now becomes active for the process of transcription to be carried out. To start the process of transcription, we have different types of polymerases are there. As in the process of replication, we studied the process to be started with the help of different types of primers. But in the transcription in prokaryotes, we studied a basic type of transcription factor, that is the polymerase. Same is the case in the transcription in eukaryotes. We have different types of RNA to be synthesized. So RNA polymerase 1 is going to be involved for the synthesis of all types of ribosomal RNAs. RNA polymerase 3 is responsible to form transfer RNA, while we are mainly concerned with our messenger RNA. So this messenger RNA is going to be synthesized with RNA polymerase 2. There are different types of basal components, activators and co-activators involved, which are responsible for the process of transcription to be carried out. So TF stands for transcription factor and 2 stands for type 2 of RNA polymerase. So transcription factor 2 and D is the subtype of this transcription factor 2. There are many types which are the component of basal components of RNA polymerase 2 and are involved to initiate the process of transcription and facilitate the binding of polymerase with the consensus sequences. So this the activity of TF2D is basically the helicase as well as kinase activity. Helicase is the unwinding activity, and kinase is helicase activity is done with the help of ATP energy. So what happens basically, as polymerases are there to initiate the process of transcription, TF2D is going to bind with the polymerase at the minus 30 sequences of which are present in the form of data box, which is called homogeneous box, before the start of transcription in the upstream region of the DNA. Upstream region is that region of the DNA which is responsible for the complex of transcription to be made on this upstream region, so that the process of transcription to be carried out smoothly. Upstream of this data box we have a kite box, which are 45 to 55 sequences upstream of data box, and is a clue for the polymerases, especially in the case of TF2D, that there are the same consensus sequences are present, and we are now going to start the process of transcription a few base pairs apart. So basically what happens, this is a complex of transcription factory. Here you can see the green one is the transcription polymerase type 2, and TF2D is responsible to bind with the polymerase 2, which like the same what happens in the prokaryotic system is going to bind with the DNA and causes the formation of a replication bubbles. Along with these basal components, we have many activators and co-activators in the form of SP1, CTF, and many others, which are, if present, are a stronger clue for the initiation of transcription, and if not, transcription will be carried out anyways. We are going to start the binding of polymerase 2 with the promoter regions, which are the consensus sequences and present in close proximity at the upstream region of the DNA in the form of data box, GC box, and CAT box. So DNA binding can cause an enhancer element that is far from the promoter in the linear DNA molecule to interact with the transcription initiation complex. When this initiation complex is made, then there is a CTD complex, that is the carboxy terminal repeat domain, which is responsible to call the other types of TF2H and TF2E to come along and causes the phosphorylation of this end. So the coming of TF2H along with the TF2D causes the initiation of the DNA transcription. As the transcription is initiated, we are now moving towards the second step of transcription, that is the elongation step. In the elongation, we are going to dissociate the TF2D from TF2H. So dephosphorylation causes the elongation to be carried out, causing the formation of messenger RNA, which is single-stranded from the double-stranded DNA. And termination happens when there is a repeat of TT sequences at the end of the DNA. That will be considered as the termination signals for the polymerase, which is DNA polymerase 2 in case of messenger RNA synthesis, and it causes the termination of the transcription. So after termination, we have the role of enhancers and silencers as well during all the initiation, elongation, and termination steps. Enhancer stimulates the transcription and are cis-acting DNA sequences, while silencers reduces the gene expression. So these silencers and enhancers are indirectly involved in the synthesis of transcription or RNA. After the synthesis of RNA, we have some post-transcription events or modification to be carried out on this primary transcript of mRNA. pMRNA is identical to its primary transcript hnRNA. So we have to we do not need any type of post-transcription modifications on the prokaryotic mRNA. While in eukaryotic mRNA, post-transcriptional modification occurs because mRNA have to be moved from the nucleus into the cytoplasm. What happens to this eukaryotic mRNA? Poly A tailing at the 3' end happens, and this is the termination of the transcription machinery, which prevents the 3' exonuclease activity. Also, to prevent the nuclease activity, 5' capping is done, and it also causes the recognition by the translation machinery, because we are going to study translation afterwards. Then successive methylations occurs, and after these two steps, which is called as capping and tailing, we have another event called as splicing, which is done on the eukaryotic mRNA. As eukaryotic mRNA contains both exons and introns, we have to remove these intervening sequences from the coding region, which is called as exons. The removal of introns from the exons and joining of exons together is called as the splicing. This splicing is done with the help of different types of proteins called making a spliceosome, and the formation of spliceosome is done with the help of single nucleotide ribonucleic acids, which are U1, U2, U3, U4, U5, U6 and U7. These are all uracil rich regions and causes the removal of introns from the exons, and afterwards this causes the joining of exons together. So these all small nuclear ribonucleoprotein particles, which are known as SNRs, join together one by one and causes the formation of spliceosome. And after the spliceosome formed, we have successive events takes place, and finally we are able to get the mature messenger RNA. The different SNRs function are: U1 cuts the 5' and U5 attacks the 3' site of the intron, U3 splices the ribosomal RNA, U7 splices the histone mRNA, and if there is any incorrect splicing occurs, then it leads to different types of disease. One of them is thalassemia. So these all SNRs units join together and cause the formation of spliceosome and causes the removal of intervening sequences, which are introns, from the exons. So we get the messenger RNA that is in the form of mature messenger RNA. Just like the post-transcription modifications of messenger RNA, we also see the different types of post-transcription modification to be done on the transfer RNA, which is made from the RNA polymerase type 3. Splicing also occur, and trimming of the different types, different ends of the transfer RNA occurs, as we all have studied in the first year biochemistry, in the chemistry of nucleotides, that transfer RNA structure is a clover leaf-like structure. So transfer RNA is being trimmed at the both ends of its structure to get the mature transfer RNA. And what happened when there are ribosomal RNA splicing also occurs, we will get different types of ribosomal RNA in the form of 5.8, 8 and 5 ribosomal RNA. So finally, another type of splicing, which is known as alternative splicing, leads to the formation of different tissue proteins. And mRNA for tropomyosin, for example, mRNA for tropomyosin undergoes alternative splicing and produces multiple isoforms of tropomyosin. So tissue specific proteins are produced from the primary transcript by the alternative splicing. Alternative splicing means we have different types of RNA are being made from the single messenger RNA by removing alternate units of exons according to the need of the function of the different types of proteins in different areas. At the end, we are going to discuss the different types of action of antibiotics on the polymerases. Rifampin inhibits the transcription by binding to the beta subunit of the RNA polymerase used in the treatment of tuberculosis. This is very important according to examination point of view. Then coming towards the actinomycin or actinomycin, therapeutic application in tumor chemotherapy by binding to the DNA template and interferes the RNA polymerase movement. So mechanism of action of actinomycin D is it interacts or incollates with the DNA strands. Actinomycin inhibits both DNA synthesis or RNA synthesis by blocking elongation. They interact with GC base pairs as they require the 2 amino group of the guanine for binding. Actinomycins are used as anti-cancer drugs. This is all about today's lecture, where we have completed the transcription in eukaryotes as well as post-transcriptional modifications of messenger RNA, transfer RNA and ribosomal RNA, along with the mechanism of action of different antibiotics at the level of transcription.